Okay, so after this coffee break, we will continue. And now we are going to hear Eva Reichenberg from the United States. She's coming from New York. And she will talk about prebiotics, epigenetics, and colon cancer risk. Please, Eva.
Thank you very much, Betty. Hello, everybody, and good morning. For my research exercise, I wanted to conduct a review on the interrelation between prebiotics, epigenetics, and colon cancer risk, and I was mentored by the wonderful Professor Betty Schwartz herself. The main objective of my research exercise was to better comprehend the relationship between prebiotics, epigenetic interactions, and the potential reduced risk in colon cancer prevalence. More specifically, I wanted to explore the current research regarding the prebiotic butyrate and how it acts as a histone deacetylase inhibitor. As for the structure of my review, I will first be addressing what these terms mean in their context, how each one is related to one another, and concluding with current standing of research and future possibilities in this research. As for some background terminology, colon cancer happens to be the fourth most prevalent cause of cancer death worldwide. And unfortunately, cancer is the leading cause of death worldwide, according to the WHO. What's remarkable about this notion is that it's potentially avoidable by a simple change of diet. What type of diet? One option is the intake of prebiotics, or a type of fiber. And how do they reduce this risk? By epigenetic modifications. So what is epigenetics exactly? It directly translates to above the genome. And what that means is it controls the expression of the genome but doesn't actually change the genome sequence itself. A wonderful example is the lifespan of a butterfly. Even though it retains the same genome throughout its life, it develops phenotypically different expressions based on the epigenetic expressions throughout its life. These epigenetic changes are due to very specific modifications involving DNA, histones, and/or chromatin. And this is by the addition or subtraction of either an acetyl group, methyl group, a phosphate group, ubiquitin group, etc. And here is a little example of a histone acetylation. In a more detailed example, here we see the acetyl groups added to histone tails, coiled around DNA, or the absence of these acetyl groups, the addition of acetyl groups by HAT, or histone acetyltransferase, and the removal of these acetyl groups by histone deacetylase. The acetylation, as you can see here, involves like the loosening of this coiling, while the removal of these acetyl groups leaves the coiling much more condensed, therefore repressing transcription with the acetyl group actively transcribing. The activity of these specific modifications can either be normal or abnormal. In terms of cancer, a normal activity is the tumor suppressor, p53, interacting with the cyclin-dependent kinase, which regulates cell cycle activity. And this cell cycle activity could be regulated to promote inhibition or the progression of the cell cycle. For abnormal activity, in terms of cancer, you often see the silencing of these regulating mechanisms, and this can result in uncontrolled proliferation of cells, also oncogenesis. As for normal tissue results, this is referred to as reprogramming, which we will talk about soon in embryonic development. And in terms of abnormal tissue, this is referred to as imprinting, which is an example of cancer. So as for reprogramming, this is actually an innate step of human development that involves epigenetic changes. Humans are born with all genes actually open to be transcribed. So epigenetics are responsible for encoding every part of our body very, very specifically, and it has to be done so correctly. Otherwise, we get malformations. And this is all mediated by very specific epigenetic interactions. Similar in mechanism, but very different in result, is imprinting. In abnormal tissue, the development is usually due to external influences, and that can be things like diet or environmental toxins, and they have the ability to remove or reorganize epigenetic marks, and possibly permanently. An example here is a low methyl donor diet, methyl donor example being folate, and if it's deficient in methyl donors, then there could be defective neural tube development in early embryogenesis, which is why it's recommended for mothers while pregnant to take folate or folic acid. Also related to external influences is the general gut composition. Metabolites from possibly food and microbial patterns interact with target receptors in the gut, and the recognition receptors of these patterns are expressed onto intestinal epithelial and mucosal immune cells. These induce signal cascades and therefore complementary adaptations like apoptosis or proliferation, angiogenesis, inflammation, and so on. Now, imprinting, in addition to possibly inferior gut composition, are two components of colon cancer. Epigenetic imprinting specifically results in the poor regulation of cell proliferation, and inferior gut composition lacks beneficial microbiota protecting the gut from possible proliferation. And here you can see the development of polyps in the transitional colon. Excuse me. Now to put together some of these concepts. If one is to have the intake of a Western diet, which is categorized as high saturated fat intake, minimal vegetable intake, and low fibrous food intake, this often results in obesity. Obesity also has the ability to alter the gut composition, therefore increasing the metabolic ability to harvest energy. And the over-harvesting of energy basically feeds cancer. In addition, when we also have a Western diet leading to obesity, this can lead to abnormal imprinting. Abnormal imprinting can induce the silencing of control regulation, again, leading to cancer. Also relating to cancer is cancer proliferation is angiogenesis, which in a general sense means the formation of new blood vessels. And it's also extremely important in embryogenesis and post-injury, post-workout healing. However, it also supports the tumor growth and metastasis of cancer through dozens of blood vessel networks that essentially feed these tumors. Angiogenesis is related to epigenetics, though, in the expression of and signaling of a particular factor known as vascular endothelial growth factor, or we'll shorten this as VEGF because it's pretty long. And when VEGF is overexpressed, this is what causes and sustains cancer. Here you can see the tumor cells expressing VEGF, which stimulates angiogenesis, or the growth of blood vessels, thus feeding and letting a tumor to metastasize. Thankfully, anti-angiogenesis exists, and this is through VEGF suppressing nutrients, and they induce apoptosis, they inhibit cell cycle progression, and inhibit angiogenesis itself. Here is a demonstration of a graph showing the effect of S-allocysteine, or SAC, which happens to be a garlic derivative, and its linear ability to induce apoptosis on hepatic carcinoma. Prebiotics, which happen to be the nutrient in focus today, are also VEGF suppressing nutrients. So other than suppressing angiogenic properties, they also promote healthy epithelial colon cells. They enhance bifidobacteria and lactobacilli, which are probiotics in the body, and those improve digestion, mineral absorption, and general gastrointestinal health. And most importantly, they stimulate the production of short-chain fatty acids. So in a general sense, probiotics ferment prebiotics to stimulate short-chain fatty acid. The most short-chain fatty acids in general for colon protection proliferate and differentiate the different types of probiotics in the gut in order to maintain the intestinal homeostasis. However, if cancer cells are present, some short-chain fatty acids can actually stimulate apoptosis by epigenetic silencing control, which is completely opposite to its normal state. And the most highly examined short-chain fatty acid is none other than the star of our show today, butyrate. Butyrate has the ability to help inhibit signaling pathways and promote apoptotic activity, and it does so by acting as a histone deacetylase inhibitor. Just a reminder from one of our first few slides, histone deacetylase acts to remove acetyl groups from histones, thus suppressing the activity of transcription. However, a histone deacetylase inhibitor inhibits this, allowing hyperacetylation to occur or the lack of removal of an acetyl group. As represented in my research exercise, butyrate acts as a histone deacetylator inhibitor by targeting histone or inhibiting the targeting of histones, which allows for the hyperacetylation. And as you can see by this diagram, here's the acetyl group and the loosening of the coiling, which allows these genes to be exposed and actively transcribed. And this results in many different actions that help in the reduction of colon cancer. More specifically, there is the availability of the active transcription of P53. As I mentioned earlier, in normal expression is a tumor suppressor, therefore inducing apoptosis of cancerous cells. Also, this availability for transcription can also leave it open to DNA damage, also resulting in apoptotic action. Butyrate is also directly responsible for stimulating the CDK inhibitor P21, and when P21 interacts with the CDK2 in G phase 1 of the cell cycle, it has the immediate inhibitory effect on the cell cycle, also ending any possible proliferation. And last but not least, butyrate or histone deacetylators have a direct influence on the downregulation of VEGF, thus putting angiogenesis or the feeding of tumors to a halt. So, in summary, prebiotic intake may result in the epigenetic expression of mechanisms capable of reducing colon cancer risk related actions in a general sense. As for clinical application, one of the most prominent sources of prebiotics or butyrate is through inulin, which is a non-digestible fructan extracted from vegetables, including onion, garlic, raw root vegetables, some sugar substitutes, as well as legumes, raw oats, and unrefined wheat and barley. Now I'd like to move on to possible future explorations. And given the extent of the current research, I would like to see future research explore these concepts through intergenerational epigenetics. As represented in my research exercise, this picture demonstrates a pregnant woman, Exhibit A. Oh, I do the wrong way. Her gestational fetus, as well as the fetus's gametic cells. And therefore, it's implied through consumption, possibly through a prebiotic, that intergenerationally, the mother can induce the epigenetic control of the fetus, and the fetus transgenerationally and epigenetically can pass this on to its further offspring. It's also known that maternal microbiota from maternal consumption in the gut directly influences the mammary glands during breastfeeding, as well as the microbiota in the skin and the lymph circulation, ultimately resulting in the colostrum milk microbiota. And the colostrum is the first, basically, dose of maternal milk that an infant receives. And that is responsible for colonizing in the infant gut, setting the environment for bacterial invasion in the infant gut. So if this is true, and we've already established that the intake of probiotics from the mother has the ability to influence them and their gut environment, then it can be assumed that maternal gut environment leading through breastfeeding also has the ability to influence a child. Of course, this is only speculation, which is why I think it would be worthwhile to look into this from a research perspective. So in conclusion, I really hope that the future research is able to solidify some of these theories. These theories being that colon cancer risk can be reduced by epigenetic anti-proliferation mechanisms induced by the consumption of prebiotics. Also, perhaps, colon cancer reduction applied through gestational intergenerational epigenetics by the consumption of prebiotics as well, through transgenerational epigenetics through the fetal gametic cells. Lastly, potentially the colon cancer risk reduction directly applied through the transmission of prebiotic microbiota in the mother's milk to the gut of the breastfed infant. I really hope that all of these are thoroughly investigated because of the huge beneficial effect it could have not only on colon cancer but general gastrointestinal health of both mothers and their subsequent offspring. Thank you very much.
Thank you very much, Eva, for your nice presentation. Any questions, please?
Thank you, Eva, for your beautiful presentation. So my question to you is, how do you think you can include prebiotics in our diet more effectively?
Well, other than the examples of prebiotics listed, like onions and garlic and unrefined wheat barley, additionally, probiotics should be ingested because, as I represented, probiotics are responsible for fermenting prebiotics. They're enhancing the stimulation of the short-chain fatty acids, like butyrate, and inducing the effects of the histone deacetylase activity. So it's while consuming probiotics and prebiotics in general are beneficial for the gut microbiota and gastrointestinal health, specifically to colon cancer, it's very important to have both because they interact with each other.
Well, talking about this interaction, I wanted to ask you if there are specific probiotics or prebiotics that interact the same way all the time. Like, is this interaction, regardless which one, is the same way all the time, or do you know about specific cases?
So prebiotics and probiotics interact in what's called like a symbiotic relationship. And the symbiotic relationship is not always equal. at least from the prebiotics perspective. So like if you have a prebiotic, like an oligosaccharide, a general prebiotic, when it interacts with the probiotic bifidobacteria, it has a favorable effect, but when it interacts with lactobacilli, it actually doesn't have a favorable effect, or doesn't have the same extent to beneficial effect as would bifidobacteria. Yeah.
Thank you, Eva, for your presentation. It was very interesting. I also wanted to know, what are some other epigenetic modifications' effect on colon cancer?
Right. So in my presentation, I focused on butyrate as a histone deacetylase inhibitor, but there are lots of different epigenetic changes that influence cancer, especially colon cancer. A really big one being DNA methylation, and that can be both the hypermethylation or the hypo, so like the presence of a methylation or the taking away of one abnormally. For hypomethylation, this influences chromatin mutations and the subsequent loosening of the chromatin, which, like histone deacetylase inhibitors, allows for transcriptional activation. So if this is unfavorable, that's a possible result. And for hypermethylation, it's involved in the mutation of P53, which is the tumor suppressor that I spoke about. And if P53 is affected, then tumors are possibly allowed to proliferate. So that would be a negative induction.
Hi. I want to ask about specificity, because the acetylation is quite general process, happens in many cells. So how do you think the specific prebiotic will affect only these cells or maybe other cells? And then what is the combination between the different effects? I mean, I don't want to prevent angiogenesis anywhere. I just want it in the tumor or...
Right. Because short-chain fatty acids in the presence of cancer specifically act in an apoptotic manner, but when there is no tumor or oncogene presence, then they actually work to proliferate, like gut bacteria, to promote health homeostasis. So it's specifically programmed, I guess, to only act in like the area of a cancer and when it is present, according to current research. Hi, Anna.
Any other question?
Yeah, I have something, but it's almost connected.
My mouth is so dry.
Maybe just I need to have some clarification of maybe brief mechanism of how probiotics stimulate production of the short-chain fatty acids.
So it's actually both sort of a cycle with probiotics. So probiotics, which are innately inhabiting the gut of any person, they fermentate prebiotics, which stimulates the production of short-chain fatty acids, but also in turn, short-chain fatty acids promote the further proliferation or differentiation of probiotics. So it's sort of like a vicious circle. It sounds like a negative thing, but it's a cycle that promotes both.
Thank you, Eva. I've heard only the last slide, okay, and I have a question about it.
Sure.
How significant do you think is the effect of bacteria which are penetrating the intestinal barrier? We don't like bacteria in the blood and in the lymph, so maybe it is not the bacteria, it's less the bacteria effect, but more the effect of the immune response, which they are just increasing in the mother.
Right. If you're referring especially to breastfeeding, it's also known that negative influences from the environment, including immune responses and the immune environment of the mother, have the ability to directly influence the immune system of the child. I guess under the assumption that prebiotics and probiotics are able to maintain homeostasis within the gut, then it would be beneficial to the child. But if there's negative infiltration of infection or any other toxins into the gut bacteria, then just like it would for the mother and the baby, it would probably offset the benefits of probiotics, and that's just my guess.
Okay. Thank you.
Any other question? Okay, thank you very much, Eva. You did a really good job. And now we have Hania. Hania Maher Taha. Please come to the podium.
I'm wearing.
Hania, she's going to talk about novel nutrigenetics and nutrigenomics approach to prevent colorectal cancer, role of vitamin D, magnesium, and Brassicaceae family. Please, Hania.
Thank you, Betty. So, welcome everyone. I would like to thank you for being here and for those who are watching us online. Before starting, I would like to thank the Foundation of Marcel et Jean Catou for their funding my studies, and special thanks to Professor Betty Schwartz for supporting me in this for this research exercise. So, an overview of what I'm going to cover in this presentation is how vitamin D interacts with the polymorphism in cytochrome P and vitamin D receptor and adenomatous polyposis coli pathway, followed by magnesium and its interaction with transient receptor metastatin 7, and finally cruciferous vegetables and its interaction with glutathione S transferase. So what is polymorphism, or what is SNP? It's actually a single nucleotide, A, T, C, or G, when in the genome, when it's different in different DNAs. And as you can see in this photo, DNA molecule 1 differs from DNA molecule 2 at a single base location. And actually, this difference is only 0.01% among the population. This very small difference, or this very small percentage, is the one that is bringing all the differences between the genetic population, genetic differences. You need to know what is nutrigenetics and nutrigenomics. In nutrigenetics is the study that identifies the influence of genetic variation and inheritance on the nutrient response and uptake, on metabolism and health. On the other hand, the complementary study, nutrigenomics, it investigates the interaction between nutrients on genes and their effect on transcriptional activity, genetic expression, and human health as a whole. So we can look at nutrigenetics and nutrigenomics as two sides of a coin, where you need to consider both the effect of genotype on nutrition and the effect of nutrients on genotype in order to perform a personalized diet. And this is the nutrition revolution, or the future of nutrition. So the objective of this presentation is to point out how do these nutrients and these genes interact together in order to protect against a common type of cancer, which is the colorectal cancer. Before I start talking about colorectal cancer, I would like to show you some facts, where every 3.5 minutes someone is diagnosed with colorectal cancer, and every 9 minutes someone dies from colorectal cancer. Actually, this data was true in 2007, and we expect that by now, more people have been diagnosed with colorectal cancer. This graph has been taken from the Office for National Statistics in England 2008, where it shows us the incidence and mortality rates, how they differ with gender and over time. As you can see for the upper two lines, the incidence of males is higher than the incidence of females. Well, this is good news for us, not for you. But the bad news here is that the lines are still going up even after introducing screening in 2006. Also, the mortality rates of males is higher than females. But the good news here is that the lines are going down due to the early detection of the cancer. On the other hand, this graph shows us how the mortality and incidence rates differ with age groups, where as one ages, his incident and mortality rates increases regardless of the gender. So what is colorectal cancer and how does it develop? Actually, colorectal cancer initiates due to several mutations in genes, mainly oncogenes which speed up cell division, or tumor suppressors which induce apoptosis. Tumors can be either adenomatous polyps or hyperplastic polyps. Both are precancerous. So I'm going to start with my first section, vitamin D. Vitamin D is thought to protect against colorectal cancer. As you can see from this graph, the relative risk of colorectal cancer is decreasing while we increase the serum vitamin D. And this can occur via several specific mechanisms. The first mechanism is by the direct interaction or the direct effect of vitamin D on the phosphorylation of cyclin-dependent kinases 4 and 6. This effect will inactivate cyclin D and in turn will disrupt the G1 phase. This same effect can occur by activating B16 expression. Another way is through the activation of P21 and P27 expressions, where they inhibit both the phosphorylation of kinase 2 and the activity of cyclin E. And this will phosphorylate RB, which is a co-repressor. Another mechanism is through the production of active vitamin D, which interacts with VDR, which is vitamin D receptor, and RXR, which is retinol X receptor. And this complex will actually induces transcription activity, which not only lead to protection against cancer, but also against many diseases. And as you can see also from this picture, that vitamin D can be obtained from supplements, food exposure, or food, which then can be metabolized by the liver cells and the kidney cells through the enzymes 25-dehydroxyvitamin D and 1,25-dehydroxyvitamin D respectively. And these enzymes are encoded by several CYPs. And we expect that mutations in one or more than in one of these CYPs will affect the production of the active vitamin D, and this will lead to the effect on the vitamin D activity. So here the question is, what is the effect of VDR if there's a polymorphism in it or mutation? So an example of VDR polymorphism is the FOK1 restrictive enzyme, which is located at the 5' of the vitamin D receptor gene. This happens when the ATG allele is changed to the ACG, which gives a shorter allele, which is less efficient at activating vitamin D target genes. And as findings show us that the mutation in the FOK1 allele is significantly increasing the risk of colorectal cancer by 84%. Similar results are for the heterozygous mutated type, with increase of 51% compared to the normal or the wild type. Another example is the BSM1 polymorphism, which is located at the non-coding 3' of the VDR gene. This region is thought to be involved in mRNA stability. And before giving you the findings, I would like to note here that this time the wild type is referred to by the recessive alleles, and the mutated type is referred to by the dominant alleles. So actually, the mutated type this time is found to be a protective factor against colorectal cancer, where it gives us a significant 13% reduction in the risk when compared to the normal type, and a 10% reduction in the risk when compared to both the heterozygous mutated type and to the normal type. Actually, this can be because of the high affinity of BSM1 to vitamin D, which means that a small amount of vitamin D is associated with the risk, and we can see this here, where vitamin D, a low tertile of vitamin D and the mutated type BB are associated with a reduced risk of colorectal cancer compared to the normal type and the high tertile of vitamin D. Well, not all studies have shown significant interactions between independent polymorphisms and the VDR polymorphism and the colorectal cancer, but several studies have shown strong interactions between haplotypes and the risk, where this means that more than one mutation or more than one polymorphism in the VDR gene are associated with reduction in the risk, and this is due especially actually on the 3' receptor, 3' region, where its functional bioactivity is low. So polymorphisms in this region will allow for transcription activity. Another pathway is the APC pathway, which here we can see that the APC gene or enzyme with the other complex here will hypophosphorylate beta-catenin. Beta-catenin is a transcription factor. This happens in normal conditions, where the hyperphosphorylation of beta-catenin will lead to ubiquitination and degradation of the beta-catenin, which means that it will not reach the nucleus, it will not cause transcription. On the other hand, when we have mutation in APC gene, this will not allow the formation of this complex, which leads to the interference of Wnt protein, which will cause hypophosphorylation of beta-catenin and accumulation of beta-catenin in the nucleus, which interacts with TCF, the T cell factor, and causing transcription. This can be modified by vitamin D, where it is shown that it significantly increases the upregulation of APC and the cell-cell adhesion E-catenin expressions. Also, it's found to be significantly increase the upregulation of APC beta-catenin score by 31%, and it may decrease the promotion of the translocation of beta-catenin from nucleus and cytoplasm to the plasma membrane by 12%. Now moving to the other section, which is magnesium. Actually, we know that age higher than 50 years and high BMI are associated or are risk factors for cancer in general. And in these findings, it is shown that high intake of magnesium is significantly associated with a decreased risk of colorectal cancer, especially for those who are above 55 years. And moderate and high intake of magnesiums are associated with decreased risk of a significant decreased risk of colorectal cancer, and even for those who are overweight or obese. So we also know that calcium can compensate with magnesium for the intestinal absorption, and polymorphism in the magnesium transporter, which is called transient receptor potential myostatin 7, can affect the magnesium effect. So the magnesium activity, sorry. So we can see in this graph that those who have one or more allele, polymorphism of this allele, and high calcium to magnesium ratio have significant increase in the risk of colorectal cancer. And we can also see that there is a significant interaction between the polymorphism and the calcium-magnesium ratio. Moving to this last part of the nutrients, cruciferous vegetables, as you can see here, here are some examples of cruciferous vegetables. Now the question is, how can they benefit our health in terms of the color of the cancer? Well, cruciferous vegetables contain high ITC, which is isothiocyanate, which exerts blocking effects on initiation of carcinogens. In addition to that, the glucosinolate, which is found in the plants, contains it induces the phase 2 enzymes, like, for example, the GST, which is the glutathione S transferase. This is main metabolic pathway for inhibition or for the degradation of ITC from the body. And low activity of GST means that ITC is degraded at a slower rate, at a greater extent at the target tissues. GST-M1 and GST-T1 are isoenzymes of the GST. And when I say null genotypes, null GST genotypes, this means that I'm talking about the low activity of this enzyme. Well, studies show that those who carry the GST-M1 and GST-T1 null genotypes have a reduced 69% of colon cancer compared to when their intake of ITC is high. And those who also carry this null genotype have a 57% reduction in the risk of colorectal cancer when high intake of ITC. So in the last decade, we have many research, so many data, and nowadays we have the knowledge, we have the plan, but we lack the tools. So it is expected that in the future they will develop a simple test, like in 5 minutes you can screen for your genes, and in 5 minutes you can just get the result of your interactions, and you'll get a personalized diet. So imagine that a simple test will allow us to define the nutritional status just within hours, and it will provide us with the proper dietary management. Imagine that customizing your diet today will add years to your life later. So to sum up, high intakes of vitamin D is found to be protective against colorectal cancer for those who have SNPs in FOK1 and APC, unlike those with the BSM1 polymorphism, because we said that a small amount of vitamin D is associated with the reduction of the risk. Those who have one or more allele THR polymorphism are recommended to have low calcium-magnesium ratio and high vitamin D intake. High ITC content or high intake of cruciferous vegetables is found to reduce the risk of colorectal cancer for those who have the GST null genotypes. And in spite of the several interactions between nutrients and polymorphisms, there is still a crosstalk about the interactions between nutrients themselves and the SNPs or the polymorphisms themselves. And we need more studies to prove these interactions. Till now, we still don't know what is the effect of these interactions on colorectal cancer. Therefore, it's time for new nutritional strategies where we need to start thinking or changing the way we think about diet and about giving recommendations or the nutritional status evaluation. This is actually a recent program done by the Organization of Nutrigenomics in the European Community. It's called Food Bowl, or Food Biomarker Alliance, where they are still developing new public databases on food metabolites, software tools, and chemical libraries. So I recommend you may check this website for not for only research but also for personal interest. And I would like to end by saying you cannot choose who your parents are, but you can do something about your diet. You cannot choose your own genes, but you can choose your diet. Thank you for your attention, and questions and comments are welcome. Thank you.
Okay. We are, yes. Okay. Please.
Microphone.
Hi, do you hear me? We have a question from Prabhat, who is connecting from Denmark. Prabhat. My question to the current presenter: do we know anything whether maternal nutrition, in parentheses vitamin D, during pregnancy can affect the development of colorectal cancer later in life in offspring?
Thank you for your question. Well, I think that yes, it may affect because while the mother is pregnant, the essential period for the genes production and all the organs production happens in that period. So if the mother's intake of vitamin D is enough, this may affect the genes in the while they are performing, and it may protect against cancer in general. Thank you. Eva.
Thank you, Hania, very much. Obviously, I really liked your topic. But given that we, as you said, we don't have the specific tools for personalized medicine just yet, given what we do know about this research, how do you recommend that we currently try to reduce the risk of colorectal cancer?
Okay, thank you, Eva. Well, I think one first recommendation I would give is to test for essential vitamins and minerals in the blood, especially for those for the tests that are not routinely done, especially vitamin D, magnesium, calcium. You can test for these. This is on one hand. And on the other hand, we can still give the general recommendations of to be more physically active, to avoid smoking, to avoid alcohol drinking, to eat more fruits and vegetables, especially cruciferous vegetables, which is known to be protective against cancer. Thank you.
I have a question, Hania. Thank you very much for your presentation. It was very nice. When we're talking about this protection with vitamin D, do we know the level that we have to supplement in order to have this effect, or just with a DRI, it's going to be enough?
Well, as the studies showed that 1,000 to 2,000, there was inconsistency of the number, but from 1,000 to 2,000 international units are recommended to affect the genes or to interact with those genes. But it's also difficult to reach the upper limit of vitamin D. So why not we just expose ourselves to the sun and eat foods which is rich in vitamin D, like eggs, cod liver oil, and food enriched with vitamin D. And in the recommendation, the RDA, this amount is already included in the RDA. So you just need to take your RDA, and that's it.
Thank you.
Welcome.
Thank you, Hania. As a continuation of the question of Fiorenza, do we know, maybe there is a study, how much, at what ages those levels are more critical? For example, if now I am deficient in vitamin D, how much time will it take to develop, for example, colorectal cancer, if I have such a predisposition, and how can we and when I'm not at risk? For example, if I'm not at risk at the first minute when I corrected my vitamin D deficiency, or I have to be at the normal levels for several months, years, or something like this?
Okay, actually for your first question, which is what is the age that is important here? Well, actually I think that it's important to protect, like even though that we know that cancer may develop after 50 years old, but still this doesn't mean that we need to neglect all the years before that. Like we need to supplement ourselves or to have enough vitamin D from early ages, because the colorectal cancer doesn't happen in 1 year, or like the genes do not change or have mutation in 1 year, and this may the effect can be for several years before that. This is for the first question. And for the second part, like when are we are not at risk?
How much time takes it to protect from the deficient state up to?
I think it depends on the person. Like it depends from genes to genes.
If you are polymorphic one, you need supplement to be very, very well supplemented.
Always.
But people don't know whether they have this polymorphism or not. So I think it's from preventive point of view, we need to do that anytime early in life.
Okay, and my last question is, can you find any connection, maybe not at the level of those genes you have been speaking here, can you find any connection between vitamin D and magnesium and maybe even the vegetables?
Yes, there's
on the physiological levels in preventing of colorectal cancer?
Actually, yes, there is an interaction between vitamin D and magnesium, where vitamin D not only helps in the absorption of calcium on one hand, but it also may induce, or it may help in the absorption of magnesium. So it's also important for magnesium.
And something about estradiol, estrogen receptor, something. Do you have any association?
Well, I think yes, there is an association where it's like magnesium will be more affinite to the receptor if vitamin D is there.
Okay.
I think,
Thank you.
yeah. Thank you.
Yes, another question.
No.
No, I have a question. Does it matter what time of the day the vitamins are taken, vitamin D and magnesium? The time of the day?
Sorry, I don't think it matters. Sorry, Betty, but I don't think it matters when do you take it. Because what do you think?
I'm sure that it matters.
It matters?
It matters because we have clocks all over the world body. So, but I cannot give you the answer, but I'm sure that there is.
For vitamin D.
Yeah. And in respect to vitamin D and estrogen, we did about 10 years ago to 12 years ago, we addressed this question whether there is some correlation between vitamin D and estrogen, okay? And indeed, because postmenopausal, the women have much less calcium retention than premenopausal. And so what we found it that estrogen upregulates the transcription of vitamin D receptor directly, and it's also through epigenetics, which is through methylation. So it controls the methylation of a vitamin D receptor and then allows transcription of vitamin D receptor. So there is a direct relationship between the levels of estrogen and vitamin D receptor.
estrogen upregulates the levels of TRPM6, which are characteristic to
magnesium.
kidney and intestinal absorption of magnesium.
Right.
So this is the connective point. Estrogen is a connective point for vitamin D.
Okay. Okay. So thank you very much, Hania. Very stimulating talk. And now we are going to hear Joseph, okay? And he will talk about zinc, efficacy of zinc and vitamin A supplementation in management of diarrhea in undernourished children under 5 years in low-income countries. Please, Joseph.
So, welcome. I'm not talking in Nutrigenetics. So be relaxed. I'm grateful to Dr. Aronis for supervising this research exercise, and also indebted to the PEARS Foundation for the financial assistance to enable me pursue my studies here. My presentation is on the efficacy of using zinc and vitamin A in the management of diarrhea in undernourished children under 5 years in low-income countries. Sorry. Oops. Okay. So, diarrhea usually is just a symptom of an infection in the gastrointestinal tract, which can be because of a bacterial, a viral, or parasitic organism, which infects us in the GIT and will lead to diarrhea. It is still one of the major causes of morbidity and mortality among children worldwide, and especially in developing countries, simply because of the various causes of diarrhea are still prevalent in the developing world, like unhealthy environments, poor hygiene and sanitation. Those are still prevalent in low developing countries, so diarrhea is still prevalent there. And unfortunately, it is one still of the leading causes of all child death, attributed to like 1.4 million children die annually because of diarrhea-related diseases. So, a significant proportion of children suffering from undernutrition in developing countries often have diarrhea episodes, and the increased incidence, severity, and duration of diarrhea worsen is undernutrition. So, the thing is disorganizing me, I don't know why. It makes undernutrition worse. Okay. So, increased incidence, duration, and severity of diarrhea worsens undernutrition, and undernutrition also worsens diarrhea. So there is a vicious cycle of this. This is just an example of a study to just emphasize the deleterious effects of diarrhea and stunting. That's a meta-analysis from various countries, and you can see down here that more than 5 episodes of diarrhea below 2 years in children is responsible for 25% of all stunting. And we know the effects of stunting in children on their cognitive and mental development, and the effects in later life, especially in the productivity and development. So it is important that we address this diarrhea issue. Now, diarrhea management, if I can say, we can term it as a double-edged sword, because diarrhea is partly good, as I said, that it is a way of the body getting rid of infections, but also the effects which it brings on the body in terms of dehydration makes increases the chances of mortality. So we need to manage diarrhea, and usual management has been by using oral hydration solutions according to WHO and UNICEF guidelines. But the studies have found that this alone may not be effective in reducing the volume, duration, and the frequency of diarrhea. However, ORS, along with micronutrient supplementation, has been found to be effective in improving diarrhea outcomes. It is also found that micronutrient deficiencies like zinc and vitamin A often coexist in undernourished children, and it is also common to see undernourished children, severe undernourished children with symptoms of vitamin A deficiency. And some trials that have used vitamin A supplementation have documented improved outcomes like reduction in mortality in children with diarrhea. So, what is the overall objective of my review? Randomized clinical trials using both zinc and vitamin A in management of diarrhea have reported inconsistent findings. So there is some gap in knowledge in some studies, where some studies have shown a positive effect on the outcomes, while others still report they are not all consistent. So that creates some gap in knowledge which needs further study and review. So I took on a task to review studies that have used zinc and vitamin A supplementation in diarrhea management in undernourished children and assess its effect on incidence, duration, and severity of diarrhea. And specific objectives was to review and summarize the effects of zinc and vitamin A supplementation on incidence, severity, and duration of diarrhea, and then to compare the findings on micronutrient use and present a general assessment on the efficacy on their efficacy in diarrhea management. So the hypothesis that combined supplementation with both zinc and vitamin A, along with oral hydration solution, could have a better therapeutic effect in diarrhea management compared to supplementation with only a single micronutrient. So the methodology that I used in this involved screening, literature screening of randomized controlled placebo trials that have used zinc and vitamin A in management of diarrhea. I chose randomized controlled trials because of they are less prone to bias and confounding. And the criteria for inclusion were studies that were limited to children under 5 years. Both hospital and community studies were included, and I also looked for studies that were carried out in developing countries, and those that measured at least one of the outcomes that I'm going to discuss here, like incidence, severity, and duration of diarrhea, and also used oral hydration solution. So studies that are reviewed met that criteria. So I searched for on online databases like PubMed and Medlin, but also I visited WHO and UNICEF websites because you can get specific data there concerning children. And research was limited to English language. And I have a total of 22 studies that were qualified that fit into being included in the study. And 9 of these studies used the zinc supplementation. 8 of them used vitamin A supplementation, and 5 studies combined both zinc and vitamin A and reported data on 2,489 children. So I'll present some findings on the effectiveness of zinc supplementation on the incidence, duration, and severity of diarrhea. And this study aimed at evaluating the effect of zinc treatment along with ORS on duration of diarrhea and stool output. Now, in this exercise, stool output is being used as an indicator to assess severity of diarrhea. The volume of stool is indicating how severe the diarrhea is. So we are going to be, whenever you will see stool output, in the results it will reflect on how severe the area is. In this study had 287 children who were 3 to 36 months old, and we are followed for 14 days. Those who are below 1 year were given 15 milligrams of zinc and 30 milligrams for those who are above 12 months old. And the severity of diarrhea in this study, which, as you can see from the total volume of stool, was significantly lower in the zinc group than the placebo. Yeah, we have the placebo group, or the control group, and then the intervention group. And also the episodes of diarrhea that were lasting more than 7 days was significantly lower in the intervention group compared to the placebo. As you can see, the odds ratios are significantly lower in the intervention group than in the placebo group. Then another study aimed at determining the role of zinc supplementation inducing diarrhea morbidity in children. In 280 children, these were aged 6 to 41 months, and we were followed for 16 weeks. So here, there was also no significant difference observed in the incidence of diarrhea in the daily and weekly supplementary groups. Now here, the children were divided into 3 groups, and they wanted to investigate whether if we administer the zinc on a daily basis or on a weekly basis, there is a difference. Now, when you looking at the results, this first one is the placebo, and these two are the supplementary groups. Now there was no significant difference in the supplementary groups when given zinc on either daily basis or a weekly basis. But when the incidence was compared on a monthly basis, there was a difference in the last month of supplementation, the 4th month of supplementation, between the daily supplementary group. You can see it is lower than the weekly. And the reason attributed to this is that the mothers or the caretakers tended to forget. If you forget, for instance, you are giving weekly and you forget to administer for 2 weeks, then the quantity, the dosage which you have skipped is greater than someone who has just forgotten who is giving on a daily and he has just forgotten to give on a single day. So that was the reason that was attributed. But still here we can see no significant difference in daily and weekly administration of zinc on the incidence of diarrhea. So in summary, of the studies that have used zinc, the incidence, severity, and duration of diarrhea was significantly lower in the zinc supplementary group than the placebo, and there was no significant difference in diarrhea outcomes observed with different dosages. In the studies I reviewed, which used also different doses of zinc, there was no significant difference, and also no statistical difference was observed on the beneficial effect of giving zinc either daily or as a weekly single dose. So I'll present some studies that have used vitamin A supplements. And this one wanted to evaluate the effect of supplementation on overall incidence of diarrheal disease and respiratory tract infections. Incidentally, most of these diseases are often studied together in most studies. It is common to find a study that has addressed diarrhea and respiratory tract infections together, or even pneumonia. So, because these are infectious diseases and they are common in children. So these were also divided into 2 groups, the placebo and the intervention group, 6 to 15 months, and we are followed for 15 months. And here also, there was no significant difference in the incidence in the vitamin A supplementary group and the placebo group. So you find that severe diarrhea, there is no significant difference between the groups, and also total diarrhea, there is no significant difference in those groups that were given vitamin A and those that are not given vitamin A. Another study also wanted to investigate the effect of vitamin A supplementation on diarrhea, and acute respiratory infections. And this was a big study with 1,140 children, aged 6 to 48 months, followed up for 1 year, and those who are below 1 year were given 100 IU of vitamin A, and those 200 IU for other children. Here, there was a significant difference in the duration of diarrhea in all types of diarrhea in the placebo and the intervention group. As you can see, there is, for severe diarrhea, there was a 20% reduction or a protective effect of supplementation on the duration of diarrhea episodes. So to wrap up this, studies that have used vitamin A, that most trials showed no statistical significant difference in incidence and severity of diarrhea between the vitamin A supplementary group and the placebo. Most trials reported a statistical significant difference in duration of diarrhea episodes in the vitamin A group compared with the placebo. And there was no significant difference in a high dose of vitamin A compared to a low dose on overall effect of diarrhea outcomes. So then I had to look for studies that have combined, administered the supplements both to the same groups. And here, I have one study that I chose to include. And here, it had to compare the clinical efficacy of supplementation with the zinc plus vitamin A, and also had other micronutrients in 167 children, and these were 6 to 24 months. So they were divided into 4 groups. One group was given zinc supplements. Second group was given vitamin A and all micronutrients. Third group was given zinc and vitamin A, and the fourth group was given the placebo, and they are followed for 14 days. And the results found that when you compare the outcomes, like duration of diarrhea, total stool output, and stool volume, it was significantly lower in all supplementary groups than the placebo. But specifically, if you look at group 3 which had zinc and vitamin A, the outcomes are significantly lower compared to other groups and the placebo. So to wrap up this, the overall incidence of severity and duration of diarrhea was lower in the zinc plus vitamin A group than the placebo and other groups. And the mean daily stool output after starting therapy was also significantly less in the zinc and zinc plus vitamin A groups than vitamin A and other groups. So what is the effect of these micronutrients on diarrhea? The effects of zinc and vitamin A on diarrhea is in various roles, in the electron transport, membrane integrity, and the immune functions. Zinc and vitamin A play a role in maintaining the integrity and faster regeneration of the epithelium in the intestinal tract. We know that pathogens or microorganisms in the GIT, when they multiply and produce toxins that destroy the gut mucosa. So if a child is deficient in zinc and vitamin A, the regeneration and the healing takes longer. Then zinc and vitamin A are also involved in enhancing immune responses, which limits bacteria overgrowth and early clearance from the intestinal pathogens. Then zinc has also been reported to improve absorption of water and electrolytes from the intestines. And because it enhances the reabsorption of water and electrolytes from the intestines, it results into reduced fluid permeability, so that less fluid is released into the GIT, which results into a reduction in stool volume. So, in conclusion, from my review, this study demonstrated that zinc supplementation in a daily or a weekly dose is efficient in diarrhea management in undernourished children. The study, however, failed to demonstrate the effectiveness of vitamin A supplementation alone on incidence and severity of diarrhea, but showed a positive effect on duration of diarrhea episodes. The study suggests that in undernourished children, supplementation with both zinc and vitamin A is more effective in diarrhea management than using only one micronutrient. So, for implications, we need to understand further which category of malnutrition will benefit more with the combined zinc and vitamin A. Because some studies have shown that administering vitamin A to healthy children increases diarrhea, and even mild malnourished children. So we need to study further to find out which group of malnourished children will benefit more from this, and also to find out whether which mode of supplementation is easily acceptable, whether they can be incorporated into oral hydrate solution, or we continue giving the supplements as capsules as it has been done. So thank you.
Thank you very much, Joseph. And now it's open for questions.
Thank you, Joseph, for your presentation. It was very good. I was thinking, you were talking about zinc as using it to the treatment of diarrhea. But do you think it could also be used as a preventive measurement for diarrhea, maybe making a stronger gut in order to face these microorganisms, or something that you can think of?
Yeah, the zinc is important in the immune functions. So it also helps in the body. The important thing is to help in fighting the infection is, first of all, it helps to fight the infections, but it is also important, as we said, in the management. So the issue is that in undernourished children, they always have low levels of these micronutrients like zinc, vitamin A, and others. So for them, their micronutrient status is less, but it is important also. It can help if the person is not deficient in zinc. It helps in fighting off infections and reducing the diarrhea.
There was also a slide of prevention. The graph, do you remember? It was also of prevention for several months.
I have a question. When you take zinc, it arrives to the small intestine. How much is absorbed in the small intestine, and how much of it gets to the large intestine, which is the part of the infection, mostly of the infection? Do you know?
How much in terms of
yeah, how much
figures mean?
Yeah. Because I think we need the zinc to arrive to the large intestine.
Yes.
And there will be affected, right?
Yes. Yeah, it also depends on other factors which enhance the absorption of nutrients. Mm-hmm. For instance, like studies have shown that if there is presence of, like, for instance, copper and zinc interact, and the copper blocks the absorption of zinc. Mm-hmm. So to ensure that there is an efficient flow of zinc, I think it's also important to note about nutrient interactions. But to address your question, I will not specifically the figures may not, I may not have the figures to say how much rich is, but we need just to ensure that we eliminate inhibitors of absorption and the transportation of zinc in the body. That's what I can say.
Thanks. Yes, Francis, and then Aliza.
Thank you, Joseph. I know quite a majority of us here are connected to having children here and there, maybe your own or others. You mentioned something on management of diarrhea that is to do with the ORS. Just from your own opinion, what could be the simplest way of making one if I want to use it in my home?
Locally, like if I can quote from the ORS, we know it's a mix of salt and sugar. That's how the locality can be made. But these days, these solutions are always cheap and available, but locally it is a pinch of salt and the sugar. Actually, it is the 1 level spoon of teaspoon level spoon of salt in 8 level spoons of sugar in 1 liter of clean water. That is ORS. But that one in undernourished children, we know that the standard ORS is high in sodium, and it is not really recommended. So I will not advise you to make it locally in your home to mix this salt and sugar. You may end up putting more sodium, which is not good for undernourished children.
Joseph, you focused on vitamin A and zinc.
Yes.
There are lots of vitamins and lots of minerals. Why did you specifically focus on these 2 nutrients? And do you know if there are any other nutrients that might also be beneficial in diarrhea that maybe have been less studied, etc.?
Yeah, you're right. But the reason why actually I included vitamin A is that it's one of the public health problems we have. Vitamin A deficiency is a major public health problem, and in developing countries it is worse, and even the effects of deficiency of vitamin A, as we know, are really grave. So there are other micronutrients which are important in diarrhea management, like for instance, vitamin E, those antioxidants which fight the infections. But I chose zinc and vitamin because zinc, for instance, in the diets which are common in the developing world, the phytates are common in our diets, so they block the absorption of zinc, and we are deficient. We don't usually take the consumption of animal proteins is also limited, which is rich in zinc. So that's why I just focused on these 2. But there are others, and I think we need to study about them.
No other questions, so we will go into the break, in the lunch break, and we will be back.